I'm a clinician scientist, work as a professor of ophthalmology in Cardiff University and at the Cardiff and Vale University Hospitals Trust Wales. And I've been here for nearly 20 years, which seems remarkable. I think my journey really did begin when I applied to do a PhD at UCL at the Institute of Ophthalmology many years ago. And this was a project in genetics, which struck me as most interesting straight away because I was really interested in genetics already back in 1995 or 1998 when this all came about. And it was working essentially to try and understand the genetics behind inherited optic atrophies or optic neuropathies, as they're sometimes called. And I was really interested in this because it was a project right from the start, collecting information about the families, what was wrong with them, so assessing their vision, looking at their optic nerves, imaging the back of the eye, taking visual field assessments and doing what's called psychophysics, where you try to probe their vision by using different markers such as fields and light responses, electrophysiology. And that was great. That was all patient related. But there was another part which was really exciting, which was collecting blood and extracting DNA and then looking at how these families passed on a particular condition, which was called dominant optic atrophy. And this is really how it all started for me, because I was given an opportunity in a way to go back to university at a time when I was already a clinician. And I could basically play in the lab and be a student again, which was amazing because you could really get into something completely new, completely exciting, which was really developing at the time. So back in 1995, very few genes for eye diseases had been discovered. One of my supervisors, Shomi Bhattacharya at the Institute, had been, in fact, one of the first people with Andrew Wright, who was a professor in Edinburgh, to discover the first gene for retinitis pigmentosa, so rhodopsin. He was my supervisor. He was really excellent at gene hunting, so this new discovery of genes linked to families. My other supervisor was Tony Moore, who was a professor of pediatric ophthalmology, and he was an incredible mentor and supervisor, and just a generally fantastic person to be around. So that was really, really, really inspiring. And he was also able to let me come to his clinics. So I was able to do some clinical work, clinical research, and then learn the laboratory techniques, which is a bit like learning how to cook. And it's amazing. It's, you know, you have to be one of these people who wants to try things, practical things. You can't just expect it all to work first time, and you can't expect to get it right first time. And sometimes you have to try it on your own without being shown. You just follow the instructions. And it was brilliant, really, learning all these new things about genes and linkage analysis and so on. And that project, which was my PhD project, led to a publication in the journal Nature Genetics and also the cloning of the gene for dominant optic atrophy, OPA1. And then it really made me desperate to continue in research because I had the bug, as it were. I was at Moorfields for 2 years after finishing my training, where I was first a senior research fellow and then a consultant. I also did a fellowship abroad at that point because it was really important to see research done elsewhere. And so I was very fortunate to be able to go to spend some time with somebody called Professor Paul Sieving, who was at that time at the Kellogg Eye Center in Michigan, Ann Arbor, who was also an incredible leader in ophthalmic genetics. He had discovered the gene for a retinal condition called retinoschisis, RS1, which was, again, very early gene to be discovered. So I spent some time with him there in the lab, again, doing experiments, but also seeing how they did all the different clinical testing in America and so on. And I was very lucky because he then moved to the National Eye Institute as the director of the National Eye Institute while I was with him as a fellow. So I was able to go to Bethesda, Washington to spend some time there as well. And that was really beautiful and exciting because I think one of the things you realized is that the American system has many pros and cons, as does ours, but it's very energetic. And at the time, it was really fast paced and really exciting. And you met some fantastic people, the sort of people whose papers and publications I'd often be seeing and reading and going to Arvo to see. So I felt really, really, very deeply immersed in the field. It was really, really fantastic to do that. Came back to the UK, obviously, to get a more permanent sort of position, and got a fellowship from the MRC. And this MRC Clinician Scientist Fellowship, which was for 5 years, allowed me to set up my own lab. And I decided that I would do that outside London so that I'd have independence, but there were many other personal becoming a senior lecturer in Cardiff. It was great because I came here to a department that was directed by somebody who was a cell biologist who'd also worked at UCL. His name was Mike Bolton, and there was a very strong cell and molecular group here, and it was really, really excellent. But nobody here had been doing clinical ophthalmic genetics. So I set up an ophthalmic genetics clinic in the University Hospital Wales, Cardiff, and see patients from all of the sort of south of Wales, and as far afield as patients would like to come, obviously. And so I love the fact that I have seen patients for many years, because I now have continuity. I've seen sometimes the same family for 20 years. And sadly, I've also seen people go from almost normal vision to being completely blind, which is not so good. But the continuity and the relationships, and also the knowledge that brings, is that it's even more important to realize that cures are needed and treatments are needed. And patients are very, very keen to know about research these days. They'll ask many questions about the latest trials, whether they'll be gene or cell therapies. Patients are increasingly anxious and frustrated because they think, there must be a trial for me out there, which isn't always true. There are more trials. And I think the next 5 to 10 years will bring a huge swathe of new clinical trials based off the research that's going on in the laboratories. My own research, apart from the patient-oriented side of things, which is more in the genetic eye disease, is retinal, but it also includes the optic nerve. So I didn't move away from my primary interest, which was the retinal ganglion cell in inherited optic neuropathies. My lab here was really designed to, at that time, make an animal model, because there were no models for the dominant optic atrophy OPA1 gene. So we designed and made a mouse model. And in fact, there was a second gene we worked on, which was called OPA3, which is very, very rare. And we made an animal mouse model for that too. We were able to study what happens in the eye, the optic nerves, the ganglion cells, which of course is very hard in patients because human tissue is really difficult to access. Animal models led us to explore how the disease mechanism works and how patients lose vision from death of retinal ganglion cells. And I always say to people that the retinal ganglion cell is what it says on the tin. It is actually in the retina, down the optic nerve. So while these might be diseases where there's optic nerve atrophy, there are actually changes in the retina first. And that's actually one of our breakthrough discoveries, which we found that we could see changes well before the vision was severely compromised in these little retinal ganglion cell projections called dendrites in the retina. And we could see that in the animal model reproducibly. And that meant that if you could intervene at an earlier stage, when the ganglion cell hadn't died yet and the patient hadn't, or the mouse hadn't gone blind yet, you might actually be able to save sight. So the idea is to now work on treatments that we could apply to either prolong sight or reverse the vision loss. Now moved away from using the mouse models. Our current focus, even for the last 3 to 4 years, has been to use human-derived skin cells, which are then transformed into pluripotent stem cells from the skin from patients, which are then differentiated or grown in the lab so that they can follow a pathway whereby they start to look like retinal ganglion cells in a dish. So our current project is not on dominant optic atrophy, but another optic atrophy, which is devastating in its effects, which is Leber's hereditary optic neuropathy, LHON. This is a condition which can lead to a sudden, completely unpredictable onset of loss of vision, first in one eye, going from normal vision down to literally just perhaps even counting fingers or hand movements over a space of 3-4 months. And then the second eye often follows 2 or 3 months, 4 months later. So a young individual, often in their late teens, early 20s, might go from a normal vision to very poor vision very rapidly, with a very large blind spot in the center of their vision called a scotoma, which means that they can not see faces. So looking at you, I would not see your eyes, your nose, your mouth. I might just see the top of your hair and your shoulders. Everything else inside that would be a blur, but they retain pretty decent vision on the periphery, on the edges. But it means obviously close, fine, detailed work is very, very challenging. Everything has to be enlarged. Patients struggle obviously initially with this devastating diagnosis, which is very, very traumatic and psychologically upsetting because it's so rapid. We now know that it isn't just men that get this, it's more often men, but it's men and women. And it's not even just late teens and early 20s. I'm seeing patients with the condition who are first losing vision in their 40s or even 50s. So we now understand the disease much more. We're picking up more and more patients with this. So it's still completely untreatable, but there are some trials. The work is funded by Fight for Sight as a project grant, allowing us to culture the cells from patients with 1 or 2 of these different mutations, and we'll then look at the ganglion cells and understand why they die. So to lay the basis of that, actually, there is a sort of a framework. The grant is really looking at the paradox of how is it possible for patients to lose so many of their retinal ganglion cells and apparently go on this journey of from normal sight to poor vision, and yet in some individuals there's actually some recovery. So we know that you don't regrow retinal ganglion cells in the eye. So what must be happening is those that didn't die were very, very sick for a time, anything up to a year or 2. They were not functioning fully. They weren't literally firing on all cylinders, and they are then able to recover. So what we know is that the underlying mutations in this disease, it's a mitochondrial mutation causing a loss of energy production in the cell, in the mitochondria, and the DNA is abnormal for that. And as a result, patients' retinal ganglion cells don't function properly. They don't make normal energy. They don't metabolize oxygen. They don't burn the sugar correctly. They may produce more reactive oxygen species. They get poisoned, they die. But some do not die. And that's what we're really exploring, because to be able to rescue those, that's the insight into some sort of recovery. So in our cells in culture, we're taking them from patients from one of the mutations where there is good recovery in some patients. And when I say recovery, it's never going to be back to normal, but it can improve. And we are exploring why the cells from the patient that did improve and the cells from a patient with the same mutation that did not improve over a period of 5 years or more, how they differ in their biochemistry and in their physiology, if you like, their cell biology. And that will enable us to pinpoint what we could do to intervene at an earlier point to encourage more of the cells to recover and to resume their normal activity in the retina. Obviously, this method would have to be applied to an individual patient at a relatively early stage of their vision loss, we think, and it might not lead to a full recovery of all ganglion cells. Absolutely, without doubt, that would be unlikely. But to improve the vision from just counting fingers or hand movements to reading halfway down the chart would be absolutely transformative. Firstly, we're looking at their biochemistry, how they make energy, what they look like, how they connect with other cells. We grow them in the dish, or we're exploring the sort of features that allow us to see why the ones that recovered are more robust. Perhaps they have defense mechanisms that the others don't. And we're also going to look through the microscopes, obviously, at their structure, how they maintain all their normal projections so that they don't end up losing their connections to other retinal ganglion cells. And we've also got a piece of equipment called a multi-electrode array, whereby we can put the cells and grow them on a little electrical grid, which is a very, very, very dense electrical grid where we can plot something like 36,000 points in literally a square centimetre. looking at their electrical signal and firing. So the cells that are better and that have recovered or that have kept growing will have better electrical activity. So this is answering the question, how is it possible that sometimes vision actually can improve and we could intervene? The history with Fight for Sight that I'm obviously very privileged to be associated with has gone back to many years ago. For 5 years, 2012 to 2017, I was on the Grant Advisory Board, which meets to assess grant applications and reviews, sometimes several hundred grants, and it's a huge task to review the grants in your section and then meet and discuss with the other colleagues. It's a whole day's endeavour, if not longer. And then from 2020 to 23, I was the vice chair of the Grant Advisory Board and on the panel again, reviewing grants, but this time, also the vice chair. And I think the relationship with Fight for Sight spanned those years, but in between, we've been able to be luckily funded by Fight for Sight with a studentship, for example. And a student that we had was looking at the role of photodynamic therapy, which is a type of light therapy, or photobiomodulation, and how that might help ganglion cells to recover. And that's actually an area of quite active research at the moment in other labs, and we're still interested in that. And I have a student who's just actually completed her PhD last week. Last Thursday, she had her PhD viva. Working on the Grant Advisory Board is immensely rewarding because you get to be able to provide guidance and reassurance perhaps to early career researchers. They can apply for small grants as well, so that they can get small amounts of funding which will allow them to kick off a project that would otherwise not be able to get going. So it spans the whole remit, really, from early career researchers through to large laboratories. And well, this whole project actually goes back to many discussions that began not long after COVID, and surprisingly long time ago, actually, whereby myself and 3 or 4 other investigators met. Initially, it was the brainchild of Andrew Dick, one of our colleagues, who we sort of had chatted together. We were all saying, in the UK, we do amazing research in vision, particularly in the retina and in the eyes, obviously. And we have fantastic centres that do world-beating research, but they're not always talking to each other and not always sharing or collaborating and not always working together. But that isn't just because we want to be nice to each other, but because the sort of synergies and the fact that you'd have larger groups working together would mean that they'd be able to achieve more, and that you'd also be able to attract more funding. Because one of the biggest problems in the vision research field, as Fight for Sight knows, because you produced a fantastic report back in, I think it's 2022, there is very little research funding in the eye. Surprisingly, and I would say for most members of the public, quite shockingly, really, because there's a huge amount of emphasis, obviously, on diseases such as dementias, cardiovascular disease, and cancer. But when you talk to people, they fear going blind just as much as they might fear some of these other things. And yet the proportion of funding going into the eye is a fraction, not exaggerating, of some of those big areas. So the idea of putting together what was really going to be a vision research institute initially, is going to be a virtual institute, was very ambitious. And the idea was we would have centres in Scotland, Northern Ireland, Wales, and England. Initially, just these people meeting to try and push it forward, but that wouldn't mean that this would be exclusive to those particular centres. And we approached not only charities like yourselves, but the UK Dementia Research Institute, DRI, for advice, because they had obviously succeeded in this initiative and got governmental funding and support. So we, with Fight for Sight's support and sponsorship, we had a meeting in Edinburgh last June, where we invited the Dementia Research Institute people and we exchanged ideas. And we also wanted to pick their brains how they had managed to put their networks together, collaborate, get funding. How did they influence government? How did they do it, basically? And then Further to this, Fight for Sight has proposed underpinning these new PhD studentships, which it's now advertised, and we're obviously recruiting applications now, which is an amazing initiative to really kickstart this, because without that kickstart, we weren't moving forward. So the generosity of Fight for Sight is provided for these additional, completely new stream of studentships, which are collaborative and multidisciplinary and should span 2 centres to encourage this real collaboration. And it's basically being called a DTP, which is along the lines of the studentships that are offered by UK research institutes, but from a charity sector. And we hope, obviously, there will be other areas in which we can advance this UK research network, because obviously Fight for Sight has been immensely generous and really supportive, but we feel we should try to also bring in other charities, and then also go to speak to the MRC and the Wellcome Trust and ask them how maybe we could work with them to create even bigger networks for early career researchers, for example, and laboratories. To go skiing very often, but I absolutely love skiing. I still go skiing, but not as often as I'd like to. But that's one of my sort of real things. I love the outdoors and I love mountains, but I love skiing as well as walking and so forth. And my other secret passion, but it won't be secret if I tell you this, is ballet. I mean, I did ballet until I was 18. I did all my grades, grade 8, and then I actually carried on a little bit at university. And recently, 5 years, I've been going to adult ballet classes and picking it all up again. And essentially, I love it. It's one of my absolute passions. It's a little bit like skiing and yoga. When you're doing ballet and you're learning new dances, you can't think about other things. So it's very good for clearing the brain. Yeah, it's really fantastic. We're connecting with a researcher who he met there, who uses the multi-electrode array. In fact, today we were exchanging some emails about the techniques.
Oh, right, okay.
So, in just sort of networking and also gaining insights into novel techniques. So I think that's really, really useful for that perspective as an early career researcher, but obviously the lab gains from it. The fantastic thing about Fight for Sight, I think, is because you have probably been one of the very few charities that has really determined to stick with it, because things happening that are close to the patients, close to the clinics. Truth is, there's no question, without the patients and their original material, whether it be their cooperation, clinical testing, and they're giving samples, without them, we wouldn't have done any of the early work. And now we really are pressing to try to find new treatments and cures, because it's been a long time. You can send a man to the moon, but obviously network is for all specialties. So ophthalmology, obviously, and then neurology and so on, which is great, because what we're trying to do is we're trying to encourage clinical trials. Now, that's mostly about clinical trials, whether it be commercial, to encourage companies to come to do the trials in the UK, or investigator-led trials. So it's really important for the economy, actually, as well, that we encourage these companies with new trials to come to the UK, because if we miss out, our patients will miss out. Because I think we are on the brink of huge developments in both delivery of genes to the eye, so gene therapies, however they're delivered, and there are going to be new waves of techniques coming through which will really expand that, as well as our understanding of vision loss and mechanisms. And also, we have a fantastic pool of data in the UK with information about these clinical diseases that will hugely underpin trials. So when we want to test new treatments, we have to understand the natural course of the disease, as it's called, the natural history. Without that, it's not possible to know that your therapy is truly effective. So we're really, in the UK, and Fight for Sight's support has really encouraged this. We're really privileged to have really good clinical resources as well as laboratory resources, and we're on the brink of big changes potentially for new treatments that are in areas where there are no treatments at the present time. Everyone in the vision community is incredibly how can I put this, inspirational. You meet patients and they really cope immensely well once they've had the opportunity to really be supported. But if you ask them what did they really, really want, it will obviously be a cure.
Yeah.
And that's what we have to be working towards. However, it seems ambitious, but that's absolutely what we have to be working. Yes, and if I think back to where we are, I went to Arvo, which is a big meeting. Time round in Arvo, listening to talks as well as doing what I was doing, I was truly struck by where we've come. We've come such a long way. Even 10 years ago, we were not talking about this huge surge of new trials and treatments, mostly, and because it's my area, but in inherited eye diseases, so retinal diseases and optic neuropathies. And it's unbelievable. It's really chalk and cheese now. Things are starting to make sense, and the numbers of possible new therapies are really big. The only danger here, of course, is this is a very, very crucial time, because these are very expensive studies, very expensive trials, very expensive research. If for some reason things don't go well, we have to work with commercial interests, with the pharma and the biotech companies. There is no way this type of work can be funded just through a fight for sight charity, or even, to be honest, universities. It has to have a commercial input. So we have to work with that, which is not a problem. I do feel that we must be much more cooperative, and
Yeah.
pharma obviously can't tell us their secret formulae, but we do need each other. They need us and we need them. There's a debate which is raging about what are clinically meaningful endpoints. So, for example, somebody who can distinguish light from dark, that's a lot. But to those individuals who can already see something on the chart, that wouldn't really be helpful. So it needs a debate. It needs a debate not only with the patients and asking them for what they believe are clinically meaningful endpoints, but it needs a debate across all the sector, really, to think about what are the best endpoints. Now, different patients with different diseases will not all fit the same mold. And also, people will be at different stages of their diseases, some early, some late. But this is also important because when it comes to appraising a new technology or a new therapy, it has to be seen as cost effective, because no society has a bottomless pit of money. And if one gene therapy trial leads to a treatment which costs £500,000 per patient, then you have to seriously start to understand what is meaningful in terms of a benefit and what is not meaningful. And this is not just raging in this country, it's raging everywhere, because in countries like the US, they don't have a bottomless pit either. So I think there's a real realization now that we need to understand what are meaningful endpoints. So patients increasingly now are taking part in questionnaire-based types of research on quality of life, and these things called PROMs and PREMs, where they're asked about, what are the important outcomes for them. And that is not just vision, but it's independence, mobility, society, and also things that psychological independence and so on. So it's a field that's really important, because again, it's lagged behind because there wasn't really anything to offer. So nobody really knew what patients would find useful and wouldn't, because there wasn't anything to offer them. So perhaps it was premature to really dig into that too much, but that now needs to be done and is being done, but it is important. If every single gene therapy was approved today, that's probably on this clinicaltrials.gov, which is a huge database of gene therapy trials, then there is no country in the world that would be able to afford it. None. I mean, it would probably be 10 times the national budget for everything. So we have to start to understand which are the best treatments, and then also we have to work to normalize the technology so that it becomes more affordable. Just want to see it as a niche treatment only available for a few that really doesn't change very much. If it's going to be meaningful, it has to be available to all of the people, and therefore it's going to have to be much, much more efficient.